the nucleus, and the cytoplasm is obviously hydrophilic. It's hydro. So. Okay. Can you touch upon ribosome production in nucleolus? Okay, what would those ribosomes be doing? Okay, so basically, the nucleolus is the location at where you're going to make your ribosome assembly, right? Once those ribosomes basically have been made, they're going to be either sent out into the intracellular fluid, right? And it's going to go floating around and basically creating proteins, or you can send those bad boys and have them attach to the rough endoplasmic reticulum and help with protein synthesis. That's basically what those ribosomes. So those ribosomes, they really aren't just going to stay in there, but the nucleolus, yes, the nucleolus is going to be helping produce ribosomes. That's the whole point of that part right there. Which I believe, if you guys look at if you look at your slide, I believe it's cellular structure PowerPoint, and you look at what is it? Slide 36 says contains DNA that controls synthesis of ribosomal RNA. So that RNA basically is what's going to then be used and converted to kind of code to make your ribosomes. So that's where it plays like an integral part in ribosome assembly. What other questions do you guys have for me? Any tips to remembering which is which for suspension and solution? In relation to what, Sean? I know you have a suspension, it's typically because you have two different molecules, two different solutions that kind of have different physical properties, and they kind of just make layers in that solution, kind of like oil and water. But what do you mean about like remembering which is which?
So on her review sheet, she goes into a definition for both. So I was just trying to see if there was any way to just kind of remember, like if you had any, because it says suspension is a heterogeneous mixture that contains solid particles sufficiently large for sedimentation, and then the solution is just a liquid mixture. So solution is just kind of like what dilutes it in a sense.
Yeah, so I would just try to remember it in terms of like the solvent being water. So if you have a solution, and let's just say we put some sugar in there, right? Not a lot of sugar. We just put a little bit of sugar, and then we mix it, and then it basically it dissolves all of that, right? That's a solution. But let's just say we put let's just say a whole bunch of oil in that water instead of sugar, and now you see two different layers. That would be a suspension.
Okay.
So the reason why they say heterogeneous is because there's more than one type of solution. There's more type of solute in there. So if it was oil and water, your two solutes would be the water and the oil. And obviously they don't mix, so that's why you'll have a suspension. Same thing will be, let's just say, if you have, what's another thing? I know sometimes they put like like food coloring. Like there'll be like food coloring with oil, and you'll see like the different colors of the oil that are suspended in different areas due to their density. That's basically what the suspension would be. So the solution is just like you actually like dissolve that in there.
Okay.
And it's uniformly distributed, as in like, you know, you mixed it well and now there's equal parts of that particle all over the solution.
Thank you.
I hope that helps, Sean.
Yeah. It did.
Okay, why does it say you need to increase the temperature to increase the reaction? What if you're already at 37 degrees Celsius? Doesn't the higher you go can cause denaturation? Yes. So it goes back to what she did for lab on Monday. I think you guys covered this. Typically increasing the temperature, it allows the average kinetic energy of that system to increase, right? So if we increase the average kinetic energy of a reaction, that means the molecules are going to also increase in speed, right? And they're going to start moving around faster. A reaction occurs when you have two molecules basically collide with each other, right? And then there's a reaction that takes place. Or if you're using an enzyme, right? For the most part, it's usually two molecules, boom, they hit each other, they react, and then either bonds are broken or something, or the bonds are formed, and then you kind of go about your day. If you go higher, right, and you get to a certain threshold where that kinetic energy is so high that it actually causes the bonds to break apart because the electrons are so excited that they just don't hold on to their bonds anymore. They kind of just go about wherever they want to go. What should we call it? Where was I going with this? This is how you know it's like too early. So yeah, so as the temperature increases, right, and those bonds start to break apart, that's when you have the denaturization, right? And then it causes the proteins to basically unravel, and then the structure no longer functions properly anymore because it's not in a certain formation, because structure dictates function, right? And then I know she mentioned with the denaturation, because I know I mentioned it too. I should have elaborated more, but denaturation it's not permanent to a certain extent. So yeah, if you go too high in the temperature, you can get to the point where it's irreversible and the protein just won't function anymore. And you just kind of like fry it. And then same thing with like base and acids. If you change the pH too drastically to a certain point where you kind of leave it in a very acidic solution or a very basic solution, then there's no way for that protein to get back to normal. But that's the reason why temperature increases the rate of a reaction, is because the reactions occur when there are molecules colliding with each other. The likelihood of those collisions increase as you increase the average kinetic energy, because now you're making those molecules move faster in that environment. So that's kind of like talking more into like the chemistry physics side of why it's going on. And then Rob just hit it on the nail with everything he just said. So I think Kaylin, because she's back, she asked a question way back. I'm gonna try to get back to it to see where she asked the question. She talked about, what did she ask? She asked something. Protein structures. That's what she asked. Okay, so what was your question about protein structures, Kaylin? can you explain lysosomes and their function a little bit more? Okay. Lysosomes, easiest way for you to remember it, at least how I remembered it when I took this class. Lysosomes are your garbage disposal. They're pretty much your garbage men, right? They basically they can break down debris in the cell. They can break down cellular parts or cells that no longer function anymore. They can get broken down in lysosomes. They have acidic environments, so they can break down bacteria and viruses and things like that. So it kind of plays a little bit of role in the immune system too. And then also you can break down food particles too. If you need to take like food particles that are in the cell that are they're a little too big to kind of break down, then you can break them down into their macromolecules and break them down from polymers into monomers a little bit easier when you put it in the lysosomes and then take it out. But that's basically what the lysosomes do. They're kind of like your garbage disposal. Yeah, because in order to get into the lysosome would have to engulf it. So yeah, phagocytosis is definitely something they would be doing. Can you go over this directly from the review sheet? Transmembrane proteins, peripheral proteins, integral membrane channel. Okay. Yeah. Well, my job's texting me right now, so yeah, we can talk about that. So let's see. Okay, so what did you want exactly to know about the peripheral proteins? Because they all have different functions, but the integral membrane proteins, just integral, right? Membrane in general, is these guys are associated with the membrane in one shape or form. So did you want to go over all of them, or?
Yeah, if we could just touch base on all of them just to kind of get a better idea.
And then to answer Rob's question really quick, they could be. They don't necessarily have to be like in both sides at the same time, but they can be. Usually integral, they usually are associated with actually being embedded in the actual plasma membrane, which is why they call them integral. I don't think these guys can move because, like I said, integral, they're integrated into the membrane. They could be facing on both sides, like Rob kind of just asked right now. But for them to actually move, the only way they're really moving is they're going to be moving kind of like in a lateral motion because the plasma membrane is a fluid type structure. It's not just rigid where it's like, okay, the protein's here. It kind of like all these things are kind of just moving around in this fluid mosaic model. I think they kind of like mentioned, right? So those proteins can shift in different locations as these things are kind of moving. But to go back to what Sean asked, so receptor is basically, that's your basic, you have a protein that's basically located in the membrane. There is some sort of substrate that goes and binds to it and basically causes some sort of reaction to take place, right? So that goes back to your whole receptor, effector, and your control center and all that stuff. So basically something binds to that receptor then causes some stimulus, some sort of response to take place. That could be sending another molecule, another protein to something else to turn on something else. It could be to open up a channel. It could be to do a number of things. It could be a receptor for a hormone. It could be for another protein to bind to it, all types of different things. So that's basically what the receptor is in a simple sense. I think you already know what an enzyme is, right? So an enzyme is going to take two substrates, put them together, cause some sort of catalytic reaction, and then release products. A channel protein is, in a simple sense, basically a protein that's embedded directly inside the membrane, and another way to call it is the transmembrane protein, and that allows things to go from the outside of the cell to the inside and from the inside to the out. One of the easiest channel proteins you can remember is aquaporins, and they say aquaporins because water goes through there, right? So obviously water can diffuse through the plasma membrane, but it's at a slow rate because remember, water is polar. The plasma membrane is polar on the outside, but what's on the inside? It's non-polar. So water can eventually make its way through, but it's kind of not efficient. So there are aquaporins that actually exist to allow larger amounts of water to go in and out of a cell. Let's see what else. Then you have the gated channel, right? So the gated channel, they call it as a gate, right? So basically, it's a solute that has to, there's a molecule that has to bind to that channel and then allow that solute or whatever that material is trying to get in to get in. So it's basically like you're getting to a security access point, right? You're like a checkpoint, and then you're basically showing your ID. And then when they look at your ID, they're like, "Oh, okay, cool, you're ready to go in," and then they'll let you go in. And the same thing for getting out. So it's like basically, like going to a security checkpoint. A cell identity marker, it's literally what it means. It's a marker, a protein marker on the cell that basically just says, "Hey, dude, I'm a part of you guys." So this is the way the body differentiates its cells from viruses and bacteria that are not supposed to be in there. If there was no way for us to identify ourselves, then our immune system would destroy our own cells. So these cell identity markers exist so that they know, okay, you're a good guy, this guy's a bad guy. And then the cell adhesion molecule, these are basically molecules that help cells to basically attach to each other. I think one of the examples they kind of go into, let me see, cell adhesion. Used in, if I'm not mistaken, I know they definitely take a role in like cell division and things like that. So, I think they call them CANs. If I'm not mistaken. But yeah, they basically help with basically binding molecules to each other and kind of structural integrity and things like that. But that's basically what cell adhesion molecules are. I hope that clarifies all of those terms. Just give me a second. I have to text my job. My other job. Okay, sorry about that, guys. Yeah, okay. Okay. Channel electricated ones. There's another one that she said is always open. Usually channel proteins are usually always open, for the most part. The gated ones are the ones that are only open when you have the molecule bind to it. And that goes into like either, it could be ligand gated, it could be voltage gated, it could be, I think it's ligand, voltage, and there's another one I can't remember off the top of my head, but those only open in relation to either a change in the cell voltage. It could be a ligand, which basically just binds to it and causes that channel to open. But channel protein is just a normal one, or a transmembrane protein. Typically, those are always open. So, like an aquaporin, those for the most part are kind of always open because the water is moving due to osmosis in and out constantly. Let's see. In one of her flashcards, she says that peripheral membranes are associated with the intracellular surface of the cell membrane. But side 12 in cellular structure. Okay, slide 12. Let me go to that. Intracellular surface. And she says are only associated with the intracellular surface of the cell membrane. Yeah, I see what you're saying. No, you're right. Let me see. Yeah, unlike men, do not enter the hydrophobic cells. Okay, it's fine. Only temporarily penetrate peripheral regions of the biolayer. But whether it's in or out, let's see. Yeah, I don't see why it wouldn't just be. I would think that it could be both sides, to be honest. Well, not ask her if it was a job, kids, you know. Yeah, you know what? Yeah, I would just email her. I don't want to be the reason why you get a question wrong. So I think there should be parts where it can be on the outside or the inside because it's again it's an integral, not an integral, but it's a membrane bound protein. So that peripheral protein is bound to the lipid layer of or the hydrophobic heads, like it's integrated in there, those peripheral proteins. But I don't see why it wouldn't be that it could be on the top layer on the outside or the bottom layer on the inside. But again, you know, I could be wrong. So I would definitely ask her just for clarification. What other questions do you guys have?
It may sound like a silly question, but any test taking tips? Because with this test, we aren't allowed to go back to any of the questions. So it's like you have to answer it and you can't skip it.
Well, I think the first thing is you have to read the question and just try to realize what the question's asking first, right? So before you, because I know we tend to have a tendency when we're under time pressure that you'll skim a question really quick. You'll see like a keyword and then you'll be like, oh, okay, that's what this means. And then you kind of skip to it. That's exactly how you get questions wrong in the test. So I think that the best thing to do is to take a good 5 seconds to actually read the question and then sit there and be like, okay, what in the world is she actually asking me about? And then when you kind of know what she's asking you about, then you go through your thought process and try to figure out, okay, what do I know that can answer that question? But if you kind of just are like in a time crunch and you're just like, oh, okay, all right, I see peripheral protein. Okay, it has to do something with that. Not necessarily. It could be talking about peripheral proteins. That doesn't necessarily mean that the question it's asking has to do with peripheral proteins. I don't know if that makes sense.
No, it does. What she was telling us was that she was going to try to trick us, like with questions. So, and she said she was going to make them wordy. So that's what scares me, are the wordy questions.
Yeah, so with wordy questions, really, that's where you just have to take your time, right? So you may think in the short term, skimming through that is going to save you time, but in the long term, you're going to lose more time because then you're going to skim through it, you're going to think that was the question, and then you're going to reread it again, and then you're going to reread it again, and then you lost already 1, 2, 3 minutes on that question when you could have read it slowly the first time, and then now you know exactly what it's asking, and you don't have to keep rereading the question, and then now you can answer it. But skimming it is only going to just cause you to just reread it again, and now you're losing seconds, minutes, and now that's precious time that's counting against you in the test. Yeah. So it's counterintuitive to skim the question. Right. But that's easier said than done because I've already been through all this, right? So obviously everyone kind of has that like innate habit of when you're kind of in time, it kind of rushes you, and then you skim through it, and then you're like, oh, okay, well, yeah, oh no, yeah, no, this is what it really means, and then you get the question, and you're like, oh, I knew the question, but I skimmed through it, you know. So to prevent that from losing easy points, read the question slowly the first time. That will save you time in the long term. So that's just me speaking from experience.
I appreciate that. I know with my experience, like with tests, I always second guess myself. So this kind of helps, like not having to go back to the question, because then you're not like going back and erasing or going, oh wait, could it be this answer?
Right. So the typical advice that I give is, if you're answering the question, you've read the question, you answered it to the best of your ability, right? Assuming you've studied, you did the best you could, right? Whatever powers that may be. Typically, the first answer you put is more likely to be correct than to be wrong. So I tell people all the time, even if you guys are going back to school in the fall and you're taking in-person exams, whatever it is, if you put the question right, you put it down, your choice, no matter what, unless you know you are 120% sure, right, without a shadow of a doubt, you got to be able to answer that question. And if you go back to it, unless you're willing to say, okay, I can bet my car on this question choice that I'm going to change, that is going to be right. If you don't feel comfortable with saying, oh, I would give my car to somebody if I'm changing this answer and I say this is the right answer, then don't change the answer. Because most of the time, you're going to get it wrong if you change your answer again. So that was mistakes that I made in my early undergrad career, and then I rectified that, and then from there on out, very rarely would I ever change a question. Very rarely. And the time that I did change the question, I got it wrong. So that's just kind of food for thought. Yeah, I would never bet my Mustang. I would never bet my Mustang on changing a question. I would just be like, all right, it is what it is. If I get it right, if I get it right. If I don't, I don't. So I think that's kind of like, yeah, so I could think that's just like kind of like something that you have to keep in mind. Another thing too is also, since you guys are going to have a time test and you are going to be under a time crunch and you can't really go back, right? Because she's going to be locking the test and it's only just answer and keep going. If you are in a situation where you've already spent a certain amount of time in this question, take your best guess and move. Don't get stuck in looking at this question forever and then lose all this time that you have for other questions, right? Because whatever, you guys are going to have whatever 50 questions, whatever it is, right? You got to think. You got to think strategically. Okay, there's this one question that's really bothering me and I can't answer it. I've already spent 2 minutes reading this thing over and over again. I don't get it. It's not working. I don't know what the answer is. Take a guess and move on. That's it, right? So just move on. You're thinking statistically, if you spent all that time, you lose the time for the future questions that now you're forced to guess, right? If you're in a time crunch, and now you're going to lose those potential points that you would have gotten right, let's just say. Mm-hmm. So is it worth it to lose 2 points because of one question and then lose 10 points down the line because now you have to rush? So you have to think about that too. So these are the types of things you think about. You're like, okay, well, is this one question worth the 10 questions I still have to answer? The answer is no, because they're all equal amount. It doesn't matter if it's an easy or a hard question. They're all equal. They all count 2 points, 3 points per question, whatever it is that she has as her grade. So that's another thing. That's mistakes that people make. They spend too much time on one question, and it's like that question is just as much as the other questions. So spend your time on the other questions. So that's what's going to kill you, is if you spend all that time on that one question's worth 2 points, but you have 5 more questions to go, those are 10 points. Which one's more important? And you don't know if those other questions would have been easy ones. So that's why, you have to just pace yourself and don't get stuck in it, right? You're not going to know everything, right? Nobody knows everything. Yeah, everybody wants to get 100 on the test. For some people, that's just not going to be possible. So you have to deal with the cards you're dealt, maximize your time, and do the best you can without wasting the time on questions that are going to count the same anyways, right? I get it if it was, let's just say, a short answer question and it was worth 10 points, then okay, yeah, you're going to spend more time on that. But these are all just going to be multiple choice. You're going to be going through it, right? There's going to be some short answer maybe. I don't know what her exam format is, but whichever ones are worth more points, then yeah, I can justify spending more time on that. But if they're all the same amount, then why are you wasting 10 minutes on one question when you have all these other questions you could be answering and they're all the same? Just doesn't make sense to me. And that's why, at least that's what I've seen when people get their test graded. That's what kind of kills them, is they get lost in one question, and then all these other questions they're forced to crunch through, and they're not thinking properly because they're skimming through it, and now they end up with a 60 on the test. And to answer Caleb's question, I drive a 2016 Ford banger. I wanted to get the GT, but it was just it was too much money in insurance, and I couldn't justify paying an extra $5K for a V8 when I'm just going to be using my Mustang as a daily. So eventually, I will get a Shelby, and I'll probably get a GT350. But for now, I'm pretty happy with my. No, I have a coupe, so I got a 2016 premium coupe. Yeah, the convertibles. I know those are more money because they have the machinery. So, are there any other questions that you guys have? Because I know you guys are probably like fried. It's only 9 a.m. Rent is especially high in New York, which is why I'm going to Philadelphia. Yeah, I mean, if I wasn't working 6 days a week, then I would do it at a more normal time. But this is kind of like the time crunch that I have, is the mornings. Not a problem. Do I know how the lab practical is going to be like? Yeah, she's going to use a lot of diagrams. There's going to be a lot of pictures, diagrams. She's definitely going to be talking about big concepts like what's one? I know she's definitely going to ask multiple questions on, the enzymes, and like their activation energy and all that stuff, and probably showing you like a chart activation and then like denaturization, all that stuff. Those are perennial favorites in any lab practical. So definitely know that stuff. Is she making a review sheet for you guys for the lab practical, or I'm not sure what she's doing with that. Okay. I may or may not be able to go on that review. I'll try to make it, but if I'm working, then it probably won't be. But yeah, it would. I would say the lab practical is big concepts with a couple of minute questions kind of sprinkled in there. But for the most part, she's going to be asking big theme questions. And if you guys went over the microscope, right? So that's definitely going to be questions she's going to have in the test. She will ask you parts in the microscope and their function for sure. That's worthy of 2-3 questions at least. Yeah, so. Okay, all right. So you guys don't know about a sheet yet. Yeah, she probably will do a review, and she might make like a quick review sheet for you guys, kind of just as like a template for topics. Any chance you know any nurse anesthesists on Long Island who would be able to be shadowed for a day or even shadowed for a single patient? I can try to ask some people, but right now your chances are slim to none because of COVID-19. I mean, even students right now that are in clinical programs are having difficulties with rotations because of this thing. So, unfortunately, shadowing is kind of on the bottom of the totem pole for any type of profession, not just like nurse anesthesias. So, I know shadowing has been suspended for everybody pretty much until further notice for now. But I mean, I could try to see if I can find anybody that knows nurse anesthesists. Um, it's probably going to be difficult to get in unless you know somebody in a hospital because nurse anesthesists typically work in the OR, and the OR is a highly regulated area. It was probably the most difficult thing for me to do to try to shadow a perfusionist in the OR. Um, but it was just knocking on a lot of doors and trying to just talk to people and just really trying to be persistent. I think is the best advice I can give you. You have to be persistent. Like you just got to be asking people over and over and over again, because obviously there's people busy, they're working full time. Um, they're thinking about, oh, okay, well, the student, okay, yeah, I'll get back to the student, I'll get back to the student, and they never get back to you. So you got to be persistent. Email them, obviously in a nice way without being too overbearing. But yeah, that's exactly why they're so stringent on it. Not so much, especially when you're talking about the OR, it's not so much about HIPAA violations, although that is something that's important, but it's more about liability. You might cause an issue in the OR, you might trip, fall, you hurt yourself, that's a liability in the hospital, all kinds of stuff can happen. So um, you don't want to be stuck in a situation where, you know, you have a student here that got hurt and now they're in your property and now you sue them because, oh man, I tripped and I hurt myself in the OR, and now the hospital's like, all right, well, we should never let you shadow in the first place. So yeah, it's just a whole bunch of issues. So typically you get in there, it's because you know somebody. That's usually the way you can get in there. Yeah, so I would probably start with your physicians that you know. Physicians always know nurses. They always know people. They're connected with people in all aspects of medicine. So I would start with the physicians that you know that you go to. Ask them to see if they know anybody. Ask your parents, and then you can go on LinkedIn and you can reach out to people because I know that they have professional accounts too, and you can reach out to nurse anesthesists that way too. Call the hospitals, go into the volunteer office and see if they can hook you up with a volunteering. Most of the time they don't because they kind of are just like, oh, well, you have to know somebody and then we can contact them for you. So, I mean, that's what I had to go through. I had to jump a whole bunch of hoops just to try to get a perfusionist to let me shadow. It was probably harder than shadowing a physician, much harder than shadowing a physician. Yeah. Because I was at one point the whole med school route and I had everything ready. I was going to take my NCAT, but I just got cold feet and I didn't feel